Well, hello everyone. Welcome and thanks for joining us for a discussion on the state of play in the vector arms race, perspective on the future of gene delivery. My name is Amber Tong and I'm an editor at Endpoints News. Now, for everyone who's watching this panel, no matter where you stand on your choice of vector, I'm guessing it starts from the same place. You have patients in mind with a genetic defect and you want to deliver a gene to fix that. But depending on what gene it is, where you want it, how much of it you need, and many other questions, you end up making different decisions. So while it's much more fun to pit rival approaches against each other, in this coming hour, I was hoping to construct this mental Venn diagram together so that we can get a clearer picture on where the overlaps are between different approaches and where they're charting their own paths. Unfortunately, I won't be the one doing the heavy lifting. Joining me today are CEOs of 3 leading gene therapy companies, each representing a unique technology for gene delivery. First, we have Asa Abeliovich from Prevail Therapeutics, which is working on adeno-associated viruses, or AAV. Then we have Geoff MacKay from AVROBIO, which is developing lentiviral-based treatments. And we'll call him AVROGeoff for the panel because we also have Geoff McDonough from Generation Bio, whom we'll call Generation Geoff, telling us about their new class of non-viral gene therapies. So to start us off, since technologies are evolving every day and AAV or lenti or non-viral delivery today isn't what it was even a year ago, I wanted to invite our panelists to bring us up to speed to talk about where your platform technology is now, what you can do with it, and for a couple of you, what you have learned from your clinical data. So why don't we start off with AVROGeoff, who's joining us from Boston.
Okay, thank you, Amber. So I'll make some general lenti points before I get into Avro. I have to say that I think that lenti is in a really good spot today, and we can begin by saying that lentiviral gene therapies are beginning to be validated across a number of unique clinical indications. And if we focus in rare disease, because of course we could roll in CAR-Ts, which would add thousands of patients to the mix. But if you limit the discussion to monogenic disease, I still think that we can begin to make some general statements. Is first of all, transformative and in some cases curative long-term efficacy has been shown. And just without getting into all of the data, I think we're all aware of multiple inherited blood disorder indications, which are either close to or have been approved, multiple primary immunodeficiencies, and a growing list of other monogenic diseases like Wiskott-Aldrich or MLD. And the track record is pretty impressive. And again, carving out oncology, we estimate that there's now over 350 patients, and we've recently added that up to be well over 1,000 patient years of data with ex vivo lenti gene therapy. And so I think you can begin to say that we're at a point where we can say with confidence that it is translating into durable efficacy. And in terms of what have been the learnings to get here, I think one that I would call out, which is kind of part of what's fun about building and managing a lenti company, is that the R&D sometimes feels like you're the pilot of a plane with only a few gauges in the cockpit. And in our world, that really translates into vector copy number, transduction efficiency, the cell dose, and the intensity of conditioning. And in the early academic days, sometimes some of those gauges weren't right. And some of the early inherited blood disorder data, for example, didn't translate beautifully. But what really is emerging as a picture is that if you can just get those 4 gauges right, a decent enough VCN, transduction, cell dose, and conditioning, it is translated in an incredibly consistent way to durable effectiveness. So I think that's sort of the general state. Now, how does Avro fit into that? I think lenti's interesting because there's not a million players in lenti. I mean, there's groups like Bluebird Bio and ourselves and Rocket and Orchard. What we've done to date is we're early in our journey, but we have dosed 12 patients across 4 separate clinical trials. And so we're certainly not at the point where we can draw real heavy concrete conclusions. But if we do interpret our data, we can say that so far, 12 out of 12 patients are showing what we would hope to see, which is a pretty clear signal that the effect of the gene therapy is doing its job, solid efficacy, and going out as far as almost 3 years in at least one patient and a couple years in multiple patients, we're seeing the same type of durability that has already been demonstrated by others for lenti gene therapy. And the safety profile is within expectations. Most of our data, 9 out of the 12 patients, is Fabry. So that's our lead indication, and that's where the bulk of the conclusions come from. And, you know, just without getting heavy into the data, we can say that the patients that have withdrawn from ERT, which was their standard, their default, remain off. All of them that have discontinued remain off. There's a pretty solid, consistent set of biomarker data in terms of enzyme up, substrate down, really impressive kidney substrate data, which we imagine will be the primary endpoint for a registration trial. And then as we get further out, we can start to look at functional measures, and we're seeing a real stabilization of things like eGFR. So the best we can say right now is that we're certainly on track in our early trials, and AVRO is really at the point now of just trying to focus on clinical execution across these 4 trials to generate more data and continue to quantify the profile.
Great. Thanks, Geoff. Next, should we move on to Asa, who's joining us from New York? Oh, I think you're still on mute.
Thank you. And thanks so much, Amber, and great opportunity here to talk about the different options available. And we really feel like that, you know, the vector choice, the technology platform choice is very much dependent on the context and what the patients need in the different indications. And you know, Prevail's approach is, I think, clear in that regard. So our mission has been from the start to focus on developing disease-modifying gene therapies for the major neurodegenerative disorders of aging, so Parkinson's, dementia such as frontotemporal dementia, and some pipeline products that include also allied pediatric indications such as neuronopathic Gaucher disease. And so, you know, these are first and foremost, you know, the urgent unmet needs. There are no disease-modifying therapies for any of these. They're challenging in that one has to get very broad distribution throughout the CNS. And for that, in that regard, AV, and particularly AV9, has really stood out and been transformative. So, as you know, a single one-time lifetime treatment with a drug such as Zolgensma, commercial product for SMA type 1 kids from Avexis Novartis, as you know, it can be life-saving and has shown, you know, very, very clear safety as well. We estimate about a dozen or more different products, clinical studies ongoing with the AV9 technology, and well over, we believe over 500 patients have been dosed with AV9, you know, with really, really very compelling safety in patients. So, you know, so of course this is foremost to us, and we saw a need to move forward quickly. We set up about 3 years ago. There were patients out there with these diseases, and we wanted to be able to push ahead. Now, our strategy, we have a pipeline, and our strategy is really a precision medicine approach, really as you outlined. So we identified, or genes have been identified for these diseases, such as Parkinson's disease. You know, a number of causative genes have been clearly identified. For instance, with Parkinson's, we're focused on glucocerebrosidase gene, which is the single most important gene, we believe. And something like about 9 to 10% of Parkinson's patients have an underlying mutation in GBA1, and this is one of the best understood genes in the human genome. We understand the genetics exquisitely well, and we understand the biology. So we know what we need to do. We need to deliver this gene. We need to deliver it broadly to the CNS of these patients. The patients are out there, and just made sense to take a validated platform that could deliver. We deliver into the cerebrospinal fluid, as do a number of groups now using AV technologies, because that is more efficient and has appeared more effective. And across the different programs in the CNS, AVs have shown remarkable durability, probably because the cells are not turning over. And so, you know, this general profile of, you know, it's a single one-time lifetime treatment, great durability, very, you know, broad distribution, and a safety record, you know, makes it clear that for our indications, from what we're trying to achieve, AV9 as a platform really stands out. And then, you know, we're just super excited about the early data we have. We now have 2 clinical stage products. We have AV9 to deliver the glucocerebrosidase gene for patients with Parkinson's disease and GBA1 mutations. We also use that for a second pediatric indication, a rare disease called neuronopathic Gaucher disease, which is a devastating early onset disease due to the mutations in the same GBA1 gene. And then we have progranulin AV9 that we deliver for patients with frontotemporal dementia with progranulin mutations. And we do have now early data from the first couple of patients that we treated with PR001, and really thrilled about how that looks. So, so these are both patients who at baseline had undetectable levels of glucocerebrosidase enzyme activity in their CSF, as expected. And at the last time point we have, which is at this point 3 to 4 months, those levels were fully normalized, so in the middle of the normal range. And first of all, what that means is that our preclinical dose finding studies very much translated into clinical clinical data, and it really validates the platform. And then specifically for that program, we're thrilled because our preclinical data and the human genetics pointed toward a 20 to 30% of normal levels as being efficacious, and full normalization as being really upside and ideal. So again, we're thrilled, but the early days, as Geoff pointed out, and I think you know, at this point, I think we're happy about where we are with this platform. But again, it's very much specific to a given, I think, context and indication.
Yeah, thanks so much, Asa. And finally, we have Generation Geoff, who's also in Boston.
I have a feeling we're never going to move past these nicknames. They're too good. I'm cherishing them already. Amber, thank you for hosting this panel. It's really a blast to be here, and the resonance of hearing, Geoff, about your progress in Fabry, which is where I started my career at Genzyme well over a decade ago. This is the dream, you know, that patients didn't know to have, you know, at that time. So it's incredibly exciting what you're up to. And Asa, for your part, I mean, the GBA, you know, the impact of GBA, or the correlation at that time, the hypothesis that it would show up in these neuronopathic diseases more broadly was just that. It was a guess. So to see you in the clinic is truly thrilling as well. And of course, all those insights came out of the Gaucher world originally, right? So, and you know, that kind of touches on the theme of this panel. You guys are both building at the cutting edge of a field that's been maturing, you know, over 20, 30 years. And that's a really important kind of context and cycle to have in mind. I think I would say we're at the beginning of another maybe 30-year cycle, which is to try to develop a series of approaches that can allow for non-viral delivery of nucleic acid therapies. And, you know, that's really begun in the siRNA and mRNA fields, primarily using conjugate technology and LNP delivery systems. And the challenge at Generation Bio has been to figure out how to get some of the benefits of what both Geoff and Asa have been speaking about, which is the opportunity to do gene replacement in the context of the nucleus, whether in a therapeutic cell or in a native cell type. And the non-viral field has really been stymied by two primary challenges. One is how to get DNA constructs or therapeutic nucleic acid constructs into the nucleus without use of the capsid, and have them reside there over the long term and drive high levels of expression. And the second challenge has been the delivery. And systemically, how do you get these constructs to the nucleus of cells of interest? So Generation Bio was really founded around that challenge, how to build a non-viral gene therapy system that would be durable, years long per dose, redosable many times as normal drugs would be, and ultimately scalable, both to reach the global demands of rare indications, but also potentially to move beyond rare indications into quite prevalent ones. And as it's turned out, to do that, to unlock that challenge, has required novel technology in three areas that really have needed to be tightly integrated. And so the company was founded around closed-ended DNA, or SEDNA, which is a linear, double-stranded, covalently closed-ended DNA construct with ITRs derived from AAV on each end. And SEDNA solves 2 out of 3 of those challenges in an important way. First, it translocates from the cytoplasm to the nucleus by itself, so it doesn't need capsid to do that. And secondly, when it's in the nucleus, it drives long-term, we think years-long expression at very high levels. And thirdly, it's amenable to really large-scale production, so it can be made in a eukaryotic cell-based system that can be scaled up to 20,000 liters, and one that allows for SEDNA to be isolated through lysis of the producer cell lines, which gives you a high degree of granular control over quality and characterization. It turns out that those 2 solution sets are important and they're necessary, but they're not enough. And where we've really spent the last 2.5 years is in inventing a new class of lipid nanoparticle delivery system that allows us to deliver this DNA construct to target cells of interest using biologic targeting. And it turns out that that's a unique challenge that has to be solved for the delivery of DNA. It's quite discrete from what's needed in a delivery system for mRNA or siRNA. And as a consequence of that third part of the platform, it's allowed us to go after systemic targets like the liver. But because the targeting is driven by a biological ligand, it opens another dimension for us to consider bringing LNPs beyond the liver to the retina, to skeletal muscle, to oncology applications. So, while we're at a very early preclinical stage compared to where AVRO and Prevail are today, our ambition is to move in 3 dimensions. One, to deliver larger payloads, allowing us to go after novel diseases, or potentially also multi-cistronic constructs where we're delivering, for example, antibodies. To move from rare to prevalent diseases on the basis of industrial manufacturing scale. And finally, to unfold that kind of potential iteratively through a variety of tissues on the basis of this targeted delivery system. So we're really in this early stage, the front end of that 30-year journey, and we're moving from mice to monkeys, and then hope to be entering the clinic in the 22-23 timeframe.
Great. Well, thanks so much, all of you. I think that was a great introduction. We covered a lot of ground. And to get more into sort of blending or comparing and contrasting what you guys said, I was curious about the sweet spots in terms of therapeutic areas and different attributes or features of the technologies that you're using respectively. And Asa, you got into some of it, why AAV is the best vehicle right now for neurodegenerative diseases. But could you maybe go into a little more detail about the specific attributes, or even some of the perhaps concerns that others would have associated with AAV delivery? And you're on mute again.
Thanks. Absolutely. So, yeah, AV9 for the types of indications we're talking about for neurodegenerative disorders is really the only platform that we felt we could move forward with to the clinic at this point. And so first and probably foremost is this remarkable ability to biodistribute, right? So this is a naturally occurring vector that's non-pathogenic and has evolved to be able to just very broadly distribute around the CNS. And even again, a one-time intravenous injection can transduce, can deliver a gene to a majority of the cells in the CNS remarkably. And then with CSF delivery, a spinal tap essentially, the process is much more efficient and effective, which is what we do. So the first point is just the need for an efficient way to broadly distribute a biological product around the CNS. And this problem goes way beyond gene therapy. This is probably the biggest problem for developing neurological drugs for the last couple decades. Just how do you get them there, essentially? And AV9 is really, in a way, the first, I think, meaningful solution for that we think is viable in this patient population. So that's the first point. And then certainly safety is a obvious consideration here, and just the safety record of having hundreds of patients, we believe over 500 patients dosed, and particularly with local delivery, with CSF delivery, there's a very very impressive safety record across different programs out there. So I think, and then I mentioned a little bit also about the just durability. And again, it's so interesting because in the liver, there are challenges with AV, with durability, but in the CNS, probably because there's less turnover, perhaps for other reasons we can talk about, but certainly durability has been remarkable, and we've seen very clear durability consistent with other programs. So, I think, those are probably I think the key aspects here that drove us. There's absolutely limitations, right? So for instance, with AVs, you have less control into exactly which cells in the CNS we deliver to, and you may not get every type of cell. So for some indications, that's going to be a problem. But in our case, both for delivering glucocerebrosidase, GBA1 gene, or progranulin, these are broadly expressed in different cell types. So we don't exactly have that as an imperative. And also, critically important is that in both cases, you have cross-correction. You have a degree of secretion and uptake, and that's very well established. And that's very helpful for us because it means we don't have to get every single cell. And so those aspects make it okay not to be able to target specific cell types. With AAVs in general, you will get an adaptive immune response when you inject the vector to the capsid. This is transient. It lasts for a few weeks. It certainly can be well managed with immunosuppression, but the exact amount of immunosuppression is always something that has to be optimized and something we've been doing. But in general, in fact, that clearly is effective, and in fact, there's very promising studies that show that with immunosuppression through delivery, there's even the opportunity to redose, which is potentially an issue, but again, less of an issue in the CNS where you seem to have very good persistence. So I think those are some of sort of the balance here, and I think it underscores why we use different, why we're using different technologies here.
Yeah. And does either of the Geoffs do you want to chime in with the sweet spots of your?
Yeah, so I'll go first. And I think it's a bit of a backward story at Avro, is that we didn't do what many do, is identify a set of diseases and, you know, ask what is the appropriate vector. It was backwards, and it was really because myself and my two co-founders really had a lifelong heritage in the ex vivo world, you know, and so we started with a bias that our skill set was very much driven by our comfort that we felt we could build the technical skill set to advance an ex vivo lenti gene therapy company. So, you know, and back in 2015, it just seemed like the time to do it. You know, I mean, Carl and Bruce were curing cancer at UPenn, and our neighbors down the street, Bluebird, were showing curative data with ex vivo lenti. So it just felt like, okay, you know, the technology's valid, but it's a very know-how intensive field. And we happen to have some of those skill sets, and we can backfill what we don't have. So that was the premise. And then, you know, step one was open up the big book of medicine and try to find diseases that fit lenti. And, you know, to say it simply, our thought process then, and actually it's our thought process today in 2020 as well, was that the sweet spot for lenti is that when what is required is that lifelong distribution of an active protein, meaning full systemic distribution above and below the neck, including into what we call these hard to reach compartments. And the obvious one would be CNS. And we're motivated by some of the data that, you know, our colleagues at now Orchard had shown in MLD, and, you know, our neighbors Bluebird and cerebral adrenoleukodystrophy. So, you know, with that sort of as the guiding post, we actually didn't find much in the big book of medicine. So we actually got out and, you know, just trolled around academic institutions looking for collaborators where there was already a substantial amount of preclinical data that showed that, you know, these weren't science projects, these were things that were ready to translate. And in the early year or so of 2015 into 16, we just partnered with academics and rolled up assets that fit that tight profile. So by starting with that, it got us to, you know, Jeff's old genzyme indications, the lysosomal disorders, because of course it was really motivating to see that delivering an exogenous enzyme could do some good. And if you looked at the patient outcomes before ERT versus after ERT, it was dramatic. So an incredible proof of concept. And yet I think everyone would acknowledge the pharmacokinetics are poor. You know, that you have a biweekly infusion of an enzyme, and yet if you test the blood 12 hours after infusion, it's gone. It works at all because some of it gets taken up intracellularly, but the majority is processed by the liver and wasted. So we felt that that was really a nice application for ex vivo gene therapy, because if we could produce even low levels of an enzyme systemically 24/7 to bathe the cells, tissues, organs, we felt we would be at the very least providing a better pharmacokinetic delivery of an exogenous of an enzyme. And so that's really what we set out to do. And when we honed in on Fabry, we just continued. And I think that 2015 and 16 was still at that opportunistic phase where, you know, there was enough clinical validation to get excited, but still a lot of opportunity to collaborate with academics that had spent 10 or 15 years of their life getting it to the point where it was ready to hand off to a company like us. So that was our approach.
Right.
So I think just to round it out in a similar way, we were starting with a very broad platform technology approach. We had a very specific set of attributes that we wanted to create with the platform. And on the basis of those, the indication selection became quite natural. We today have 8 clinical programs or clinical targets, let's say, 5 in the liver and 3 in the retina. I'll just confine, you know, for the purposes of this answer, the perspective to the liver, which, as Asa pointed out, is a systemic target. It's an organ that remains mitotic throughout life. It's highly mitotic in childhood. So the ability to get to the right dose for all patients and to extend efficacy through redosing has particular advantages for the liver. And of course, it goes without saying, there's a whole laundry list of intrinsic liver diseases that have a genetic basis that can be addressed through broad biodistribution to hepatocytes. And also, you can use the liver as a biofactory to address systemic diseases. So it's not surprising that our 2 lead indications are PKU and hemophilia A, PKU being a great proof of concept indication for an intracellular disease, and hemophilia A being one where we can elaborate a protein for systemic. therapeutic purposes. You know, I think the sweet spot, if you will, for the application of the technology, you know, really sits at two quite opposing poles therapeutically. On one hand, one of the limitations of addressing genetic disease in the liver, particularly with AAV, is that you can really only dose once. And as a consequence, you have to pick the highest tolerated dose and give the same dose to all patients. And because the liver is so mitotic early in life, you're really forced to take that shot quite late. So in kids who are 16 years of age or older at the moment. And of course, the best day of every child's life with a genetic disease is the day they're born. And as they grow, they lose function and wellness. So our ambition on the one pole is to be able to dose all eligible kids as close to birth as possible, to repeat dosing every 3 weeks using biomarkers until we know that their individualized level of expression is appropriate, and then to boost their doses as the liver grows through grade school, and in the most aspirational case, send those kids to college with little connection to the disease they were born with. And I think a redosable, titratable liver nucleic acids to the nucleus has that potential. The other pole that we're really interested in and playing for is just this broad concept, as Geoff was saying, of allowing the body to elaborate its own proteins for therapeutic benefit. And you know, our sense is that in the very largest indications, using the liver as a biofactory could actually even extend the reach of current biologics. Passively manufacturing and delivering biologics to patients every 3 weeks or every 6 weeks or 3 months imposes a certain scale limitation, as opposed to being able to allow patients to make their own biologics over time. So we see, for example, delivering the genes to create antibody therapeutics to patients using our technology as a way to even expand the reach of current biologics. So from very rare, very early to, you know, much larger, more mainstream diseases, these are the two sweet spots where we hope to build on the foundation of what's there today.
Right. And so I guess with AAV, we all are sort of more familiar with what the inherent limitations are, whether it's gene size or the things that that Asa was mentioning about cell types and all. But so with lenti and non-viral, have you begun to see, I guess, some of the inherent design limitations, things that it just can't do, or areas it just isn't quite suited to go?
Well, I think that we do see two limitations, and I think, you know, that the nice thing about lenti is, you know, we have our own issues to deal with, but what we don't have to deal with are like liver toxicity issues, immunogenicity, or the, you know, the declining durability topics that have been, you know, so newsworthy recently. You know, the challenges with lenti for an integrating vector are very, very different. And, you know, I think the first one is that it's a complex business model. It is, you know, that we have to take the patient's blood, select out the patient's own unique stem cells, give their own unique stem cells an upgrade, cryopreserve the product, ship it back, you know, and so each patient is a personalized medicine experience. So that's, you know, it's manageable, but it's a logistical issue. And then the second issue relates to patient tolerability, not of the gene therapy, but of the concomitant conditioning regimen. And this is an area that, you know, as is often the case, you know, when you're seeing the really impressive set of efficacy data that is emerging in lenti, now there's just a pouring of energy units towards improving the patient experience with the concomitant conditioning regimen. And just to give some people some backdrop, is because we infuse the patient's own gene modified cells back into the patient, just prior to that, a day or two prior, we need to implement a conditioning regimen to create space, primarily in the bone marrow, for these gene modified cells to have a home to be able to adequately engraft. Because if that doesn't happen, you don't get that long-term durability. So the focus right now is how do you improve the tolerability profile of that conditioning regime? And there's immediate term answers to that, which is really, instead of what was used historically in bone marrow transplant, which is sort of a polypharmacy approach of combining cyclophosphamide with a higher dose of busulfan without AUC monitoring, and targeting patients that have already had multiple chemo rounds and patients that come into the equation with impaired bone marrow. Instead of doing that, what we're really trying to pioneer now, and seeing very good early results, is a single agent conditioning regimen, a single cycle, mid-range dose, tight AUC monitoring is half the story. And the other half of the story is targeting patients that come in to the equation with healthy liver, healthy immune system, and healthy bone marrow. And what emerges out of that equation is an adverse event profile that is, significantly improved versus, some of the conventional bone marrow outcomes of the past. So I think it's that personalized medicine, and it's continuing to push the patient tolerability forward related to the conditioning regime. And I think many of us are also aware that there are companies that are working on next generation conditioning regimes, such as Magenta and others. And of course, their success is only going to help the field go forward as well.
Yeah, I guess I would answer the question a little bit inside out. In our case, Amber, I think the field of non-viral gene delivery it's a puzzle that needs to be put together, and the minimal set is this combination of durable expression in the nucleus, redosing, and tissue-specific delivery on a basis of scalable manufacturing. And each time you put that together, it's fit for purpose for a given tissue or cell type. So for us, cracking that equation, if you will, for the liver and the retina unlocks a certain set of targets and opportunities, but we haven't cracked that set of equations to Asa's point for the CNS. That's just a very difficult thing to do, and we have lots of aspirations to do that and to unlock other tissues, but we haven't done it yet. So our goal is to have a low therapeutic hurdle to administer our platform in any given tissue, but we will expand only as and when we have a tissue or cell specific way to target and access and broadly biodistribute in each tissue. So, I think it's just about moving through a set of opportunity spaces, but it's not like they all open up, with one delivery system. The delivery system is cell specific in a given tissue, and you move one at a time.
Right. And Geoff, I wanted to go back to something you said, and I was really glad you brought it up, which is about the business model and really how you move the cell material, the gene material through the whole process. And I was curious, so for each of the technologies that you use, what's the current infrastructure like for manufacturing these vectors? Like how do you manufacture them, and what's like the cost, the speed, and really the whole picture, the intensity of it? And I guess Generation Geoff, why don't you start? Or any of you can you hear me?
Amber, I lost you at the end of your question. I thought maybe you said my name. Sorry. Can you
Oh, yes.
rewind?
Oh,
sorry.
Yeah, rewind. Sorry. Yeah. Just if you can start talk about your process at Generation, that'd be good.
Yeah, thank you. Okay, so the part where they said, please speak, Generation Geoff, is of course where I missed you. Sorry for that. I think every technology has a series of problems to solve, and those problems at the cutting edge usually are about safety and efficacy first. But I think the X factor that all of us are aiming to get to is a place where we can make our therapies as broadly available as possible. And one of the elements in that X factor, of course, is just how much it costs to make them. And that has been straightforwardly a very significant limitation for biologics to date. We are nowhere near reaching the full penetration curve of what the demand could or should be globally for biologics. And of course, we're a long way from reaching that from the perspective of gene therapy of any kind. I mean, all genetic medicines are at the front end of that curve. So I think one of the elements that we're really interested in and have invested heavily behind is the concept of non-viral production. And in one way, there's nothing very fancy about that. Cell-based production is something that's used routinely for biologics. So if you could imagine greatly reducing the effective dose of a biologic and extending its duration of action in the liver, for example, to 5 years, you can naturally see you get a very large multiplier on production capacity on a biologics basis. So, for the specific case of Generation Bio, producing Sedna in biologics infrastructure, never using sort of a viral system along the way, it gives you the same access to large-scale manufacturing as current biologics enjoy. Specifically, upstream scale up to 20,000 liters, one-to-one downstream processing, and high-flow drug product production. Our view of that all the way through the supply chain is that we'll be selecting for each indication a minimally clinically relevant dose, and then titration occurs through redosing. So there's something quite economical about that approach to both dosing and the supply chain that we hope will lead us to a place where payers can pay a more drug-like cost with a better ability to predict both durability through redosing as well as patient-specific efficacy also through redosing at the initiation of therapy. So that's kind of the ambition, and it's what the technology platform allows us to play for. But still, there's lots to do in terms of process refinement and tech transfer and scaling and all the normal things that apply to the biologics world.
So how are you doing it today, actually?
Well, we've invested over the last 3 years to essentially invent and refine the process for our specific construct, both on the drug substance and on the drug product side. So in other words, creating the nucleic acid construct, characterizing, purifying, and then ultimately formulating in the lipid nanoparticle. So we own all of the kind of know-how and intellectual property behind how to do that. And we've had the ambition to maintain control over that at a development scale and at quite an advanced research scale. But we've actively tech transferred that to GMP CMOs, a couple of them, in order to be able to access existing scale in those systems. And Asa, I'm sure you would relate to this. It's just super competitive. There's an immature base of viral manufacturing capacity globally today, and it's hard to kind of compete and get yourself in there. It's quite the opposite from the perspective of biologics capacity. So we feel good about having a distributed supply chain model now that we've been able to tech transfer the process to a couple of different CMOs on that basis. So it's a mixed internal and external model.
Yeah. So Asa, over to you for the flip side of that story.
Yeah. Thanks. No, absolutely. This is a huge focus, and particularly for us, in part because it is a place that people have been tripped up along the way, and in part because we are interested in what are ultimately some really large applications. Gene therapy's mostly been focused on quite rare indications, but Parkinson's GBA, there's 100,000 or so Americans, right? So these are significant populations. But again, it's a critical aspect, of course, of moving forward. So we have intensively built in-house process development, analytical development over the last 2 years or so. And we actually have 2 platforms that serve different purposes. We have sort of an early to clinic platform for our phase 1/2 material that's allowed us to move. We have 3 open active INDs with 2 clinical stage products, and it's really because we have that platform. That's a HEC-293 transient transfection adherent system. It's effective, it's well established, and it's outsourceable. And so we've been pleased with that. But at the same time, we have all along the way really focused on developing a late clinical and commercial platform, and that based really on the data we've generated, we decided to focus on a baculovirus-based system, as have a number of other groups. Now, baculovirus, as you know, is a good example of some place where you really need to develop the processes in-house, as Geoff said, and this is not something that one can readily, I think, outsource. So we developed the processes, both and the analytics really for this, and have gone quite a ways now, and are completely really on track to have this platform ready when we need material for pivotal studies for both of these programs. And so the baculovirus platform does have remarkable scalability. It's more than 10x the other platforms that we've compared this to, and that's also evident from other people's experience. But beyond that, what we found is that the quality and even the scale of the comparability at scale, it really stands out with this baculovirus platform. So, and we've started to detail this. Won't go into that here necessarily, but really looks very promising. But no question, this is absolutely critical for moving forward with these programs. We have to have programs that can deliver to large patient populations.
And do you find yourself actually competing for space with the parts that are outsourceable?
Yeah, not really. So we develop the processes, we own the processes, but we then have partnered with Lonza actually for the brick and mortar GMP manufacturing, and that's worked well for us at this point. But we have the flexibility to take other approaches, and we are constantly evaluating that. This model works really well for us right now. It's allowed us to move this quickly, but absolutely, as we move forward, that's going to be an issue. But at this very point in time, we're happy with where we are.
And so, AVROBIO, for you, do you find yourself closer to either of ASA or generation?
Well, I mean, so some of the principles, I think our technology is of course different, but the idea of prioritizing process development, I think that this is really the nucleus of what we think is our core advantage, and really why we founded the company, because we really felt that the starting point was incredible science, but almost like an open manual Petri dish manufacturing process. And so the first step was really to lay out the entire value chain and ask, what are the bits that really are value add that we need or want to be the best in the world at, and what can we rely on partners? And I think that and equally importantly, what has to be done before you get into the clinic, and what can you do via these in vitro comparability process changes as we go. And of course, in our world, stem cell quality needs to be sorted up front, cryopreservation needs to be sorted up front. But then we were able to, as we got into the clinic, have pre-IND meetings and propose some process changes. And there were some critical things that we had to get in place in order to deliver on our vision of a cost-effective, highly scalable global product. And one was optimizing our vector, such that it is really tunable to manufacturing by adjusting the multiplicity of infection. We could push the VCN, and we could have a titer of 10 to the 9th. Secondly, be able to manufacture in large bioreactors. And one day this won't be considered large, but a 200 liter serum-free suspension bioreactor. So with the titer I mentioned, that means each run can produce enough drug product for treat 50 patients. And so, a single run can meet the needs of the entire Fabry clinical development program, for example. And it's pretty easy to do the math that if we have 4 suites, 12 runs per suite, 50 patients per run, you then can treat thousands of patients in a year. And if you needed to double that, go to 8 suites. So we really do believe now today with the high titer and the large bioreactors that we've turned the vector into what it always should be, which is a long lead time critical raw material. You need to think about it and plan for it, but it's not on the critical path to hold us back. But we have the second issue of manufacturing cell-based drug product. And that's really the third innovation for us, and maybe the biggest, is to be able to manufacture in a fully automated closed system. And the beauty of that is now that all of Fabry and all of Gaucher and soon the rest of our portfolio is manufactured in these small automated pods about the size of a dishwasher, they are themselves a fully enclosed clean room. So what it does is it increases scale immediately tenfold, because you could have 10 little pods in the same low-grade clean room rather than being able to only manufacture one at a time. So scale is one advantage, but maybe the more important benefit for us is we now have picked up these pods and put them in CMOs in Australia, in California, in Houston, and in a quarter or two in Europe. And they have the same proprietary AVRO algorithm. So if you hit the go button in Houston or in Melbourne, you get the same drug product out at the end. And so for us, this technological solution has enabled us to increase scale and to globalize at an early time. And, you know, of course, for biological facilities, it's if you want to manufacture someone else, you have all of those challenges of validation. Here, because it is a fully closed system, it's just a matter of doing some engineering runs to validate, and then we can produce in a different geography. So we do think that, you know, we've approached this with technological innovation as the driver, as opposed to saying, let's build the big bricks and mortar and occupy every step. So now that we've done the process development, we have the vector, we have the bioreactors, and we have the automation, we do house them in CMOs around the world.
Right. So manufacturing efficiency is certainly a big part of, I guess, driving down the cost, as Generation Geoff was alluding to. But do you all agree that it is the one of the biggest contributing factors? What are the other things that you think are driving costs, and ultimately, how long will it really take as an industry, as a field, to get prices down to a level that's fair and reasonable and affordable for patients?
Well, I mean, I could start with the fair and reasonable, because of course, what we would invite the viewer to be is rational, and to look at what the cost offset is, because of course, you know, the lifetime cost of a Fabry patient's standard of care today is $12 to $14 million. And the fact that it's biweekly infusions is one thing. So then what would be value-based? You know, I think if a gene therapy negates the need for that $12 to $14 million and isn't looking for the full cost offset of that $12 or $14 million, is looking for a small fraction of it, you know, I would think that falls into the fair and reasonable. And I think most of the field has embraced some of the methodologies of groups like ICER and NICE, which are, you know, on one hand looking at 5-year cost offsets, and then are there any other, you know, real value measures? So, you know, I think that, you know, sometimes we use the term value-based pricing. We're not asking for value-based pricing, because the value would get you up to what today's standard of care is asking, where no matter what, this will save the healthcare system an absolute fortune if we hit our target product profile of a safe, effective therapy. So I recognize society, of course, has the question, you know, are they going to be able to make the mental leap to, you know, look at the holistic costs and the potential savings? But our hope would be that there's some way to you know, have a rational solution. And I think the payers are being very productive in proposing some now.
Right. Asa, what's your take with, I mean, your AAV has currently the 2 approved gene therapies on the market. They're AAV based. So where do you see that going?
Yeah, so I'd say first of all, we're at a phase 1/2, you know, and we treated a handful of patients. So I think it really is premature, probably to talk in any detail about pricing, I think, at this point. And, you know, I think over the next 2-3 years, and as Geoff mentioned, some of this, fortunately, I think will get worked out, and look, these are the indications we're talking about. There are absolutely no treatments, and there's many patients with Parkinson's, with ALS, frontotemporal dementia, Alzheimer's. These are huge societal needs. So this will get worked out in the next 2-3 years. It'll certainly get worked out, I think, before we are, in a timely manner for us. The one other thing I would point out is that, as I said before, with AAVs and AAV technologies, these have almost universally been applied to rare and ultra rare indications, and that adds a layer of complexity, obviously, with pricing. It's obviously much more complicated to develop therapies for ultra rare indications. We're doing something really different here when we talk about Parkinson's. Even the GPA subpopulation of Parkinson's, there's 9 to 10% of Parkinson's patients in the U.S. have an underlying mutation in GPA1. So, it's 100,000 or so Americans. So the scale here is gonna matter, and is gonna be helpful, I think, also. But overall, I think it's still early days.
Right. And I guess it's even earlier days for Generation Geoff, but how, I guess by the time you go to market, how do you foresee or envision where we will be?
Yeah, I mean, I share the view of Asa and Geoff that payers will be constructive. They've already demonstrated that they want to be constructive. I think the broader question is really a societal one, right? As these technologies start to more fully meet the global need for rare diseases, and also to penetrate into prevalent diseases. The only aspect that hasn't been mentioned, I think is important to keep in mind, is payers also want security. So I think one of the biggest challenges that's been there for systemic gene therapy is not knowing how long it will last. So how do you really deal with that when you're being asked to pay a lot up front, but not knowing, whether you'll get the effect you want, and if you do get it, how long will it last? So I think security is another component. And I do think that some of these other aspects will help to alleviate some of the pressure that budget impact plays. And budget impacts has killed many appropriate paying, mechanisms and fully fair and reasonable pricing, approaches, just because of the reality of having to manage an annual budget. So, there's a theoretical part to this, and then there's just a reality of how payers work. So, I think this will be a dialogue probably as long as any of us will be in the business.
Right. So I think we're right about at the end of our allotted hour, but before we go, I thought I wanted all of you to maybe pitch in on just one big thing that remains to be done for patients on each of the platforms that you're still trying to address, like a priority or an open area. And so just, I guess, real quick, you have like less than 1 minute each. But,
yeah, so I can go first. I mean, I think that at the point in time we're at for Fabry, just as an example, we have 9 patients worth of data. We're pretty happy with it. So our thoughts are turned less to how do we get into the clinic to how do we get out of the clinic, and less about just registration, but broad label. And also to address what we just talked about, having a compelling economic health policy rationalization. So we're actually expanding our clinical development program very significantly to address label issues, not approval issues. So, young, old, male, female, mutation independent, naive, switch. So it really has had a significant shift in everything that we're doing in clinical development, because it wasn't very long ago we were just, trying to show that we're seeing an effect. And I think now we're thinking, okay, we're bullish enough on that, but now what does it look like outside the clinic, which is a very different question.
Great. I think this is a tremendously exciting time is what I'd say. I'm a neurologist and have taken care of a lot of patients with these disorders, Parkinson's, Alzheimer's, ALS, frontotemporal dementia. And what we need to do now is really move forward, is what I would say. I mean, we have, you know, we have a technology that works in terms of delivering the genes that people, you know, that these patients need. And, you know, there's nothing available. You know, it's pretty tough, right, for patients and for families having no therapies available whatsoever. And it's important for the clinicians to have sort of options as well. So that's the stage we're at. We have you know, open active INDs now, and we're screening patients and dosing. And I think we just need to move forward. We'll you know, and the very early data we have is just really exciting.
Yeah, I mean, I think the one thing, if you ask it that way, Amber, is to, you know, make real a day when getting these genes, the right genes to the right patient at the right dose repeatedly, is completely and utterly routine. That's the goal, right? And we're all approaching it in a different way and aiming for different sweet spots, but we'd like this kind of lifelong gene replacement just to become a routine reality in the way that you put gas in your car. That's what we're aiming for.
When that happens, we can hang up the cleats and go home.
Indeed, you are so right. It's been fun to be on the panel with you guys. Thank you. And Amber, thanks for bringing us together too.
Yeah, thanks.
Likewise.
Really appreciate it.
Amber.
Yeah, thank you. That's a great way to end. And really appreciate your time. All the best for what you do.
Great. Thank you. Take care.
Thank you. Take care.